Okay, good morning, everyone. My name is Reinhard van Krevel from Radboud University. I've been working with colleagues in Indonesia for a long time, for over 20 years, mostly on TB, but also on other infections. Today, we'll speak about TB and focus on the collaborative TB research between Universitas Padjadjaran in Bandung and the Radboud in Nijmegen. So we conduct patient-related research, and this started this way 20 years ago with Bhakti Alishibana, who leads the TB research in Bandung. And the first study was a study in Jakarta where we compared patients and people without TB to see if there was a genetic reason why they were susceptible to TB. Because we had to diagnose patients, we also focused on the diagnostics, and there was a second Indonesian researcher who became involved, Ida Parwati. And of course, we have to treat patients with TB, and we focused and studied the antibiotic treatment, and then became the third PhD, international PhD in this collaboration, of Nina Ruslami. And well, since then, many things have happened, many projects, since appearance of the first scientific paper, but now there's a strong group, the Bandung TB Working Group, and it's not just Nijmegen from outside that helps, also other international partners, with three aims: internationally credible and relevant research, impact on the quality of TB prevention and care, and three, building institutional and individual research capacity. So for those who are less familiar with TB, someone is maybe exposed to an infectious TB patient. That person may develop latent tuberculosis infection. 25% of the world population is probably infected with this bacterium, but they're not symptomatic and they're not contagious. But a proportion of these people will develop active TB, and that may have a certain outcome. Let's say it needs treatment, and some people die and some people survive. Well, basically in our TB research group, so one important question in this natural sequence of events is what factors drive these steps, and what makes that some people remain uninfected and others develop very severe disease, etc. The second is we're not just trying to understand, we're also trying to improve. So how can we improve patient outcome? And also how can we improve TB control at population level? And so if you think about those factors, it may be the bacterium, so it may be the virulence of the bacterium, it may be drug resistance, but it may also host immunity or comorbidity like diabetes, what we will address today, or health systems factors that impede recovery. We will also look at that today. If it's about TB control, we won't speak so much about it, but we're working and thinking about many different things, from sort of practical solutions to more sort of immunologically and tuned interventions. And how do we do this? Well, it's patient studies, it's patient cohorts, it's also sometimes clinical trials, it's diagnostic studies, but it's also lab sciences, from simple to very advanced epidemiologically and public health studies. And finally, we want to have an impact. And of course, that's not just by writing scientific papers, it's also by contributing to guidelines or education and disseminating our findings and engagement with relevant people, like the TB program in Indonesia, for instance. Well, just one outcome of this TB collaborative work over the years is international PhDs, most, not all, by Indonesian candidates. This is a Dutch PhD. And this is a Dutch PhD, and this is a Dutch PhD, and there is one New Zealand, but the others are Indonesian who conducted, at Radboud University, conducted an international PhD in this collaboration. And there's ongoing PhD projects on a variety of things. Here is the both from immunology to more public health or studies. Now, with that introduction, so today we'll just give a very brief glimpse, let's say, of what we do. And we'll start, and 3 PhDs, ongoing PhD, will present. And we'll start with Bonnie, who will speak about a TB patient's journey. So Bonnie, the floor is yours, and just tell me when you want me to move slides. Maybe you have to unmute yourself.
Okay, thank you, Pak Renat, for a very nice introduction. So my name is Bonnie Wim Lestari. So I'm from Bandung, from Universitas Padjadjaran. So I am now a PhD at Radboud Institute for Sciences since 2017. And today I'm going to share one of my work. Our work in Bandung. It's about a patient pathway to diagnosis and treatment in Indonesia. Next slide, Pak Renat. Okay, so tuberculosis in Indonesia. As we know that tuberculosis is a major public health problem worldwide with over 10 million cases and an estimated 1.1 million deaths annually. So Indonesia currently ranked 3rd in the number of TB cases globally, with an incidence that is 100-fold higher compared to the Netherlands. So compared to Africa, the prevalence of HIV among TB patients is relatively low. But on the other hand, many of our patients have diabetes as a risk factor for TB. So we found that diabetes among TB is around 13%. And for the multi-drug resistant TB, 2.8% in new TB cases and 16% in previously treated cases. Next slide, Farin. Okay, in Indonesia, TB can be managed in public and also the private sector. And according to the National TB Program, TB diagnosis should be based on sputum microscopy and treatment delivered according to the DOT strategy. DOT means direct, observe, treatment, short course. So TB microscopy and drugs are provided by the government, but diagnosis and treatment aren't available at every level. So patient can be referred between public and private sector, or can be moved vertically between primary and hospital level. So the major obstacle of TB control in Indonesia is that many TB cases remain undetected and undiagnosed. So this poses a threat for ongoing transmission. So in a survey in 2017, 1 in every 3 TB cases remain undetected. And additionally, from those detected, almost half don't get notified to the National TB Program. Okay, so to improve our understanding, next slide, Pak Renat. Yes. Yes, so to improve our understanding about these gaps in TB control, we would like to know, so what is the main question here is, what is the patient pathway to diagnosis and treatment? So by knowing this pathway, we hope we can identify where exactly this patient missed from our health system. And then to be specified, we would like to know what healthcare provider do they meet, and then how long does it take until TB diagnosis and treatment, and what factors actually account for delayed TB diagnosis and treatment. Next slide. So we conducted a cross-sectional study, Bandung, recruiting adults with newly diagnosed TB from 2017 to 2019 from community health center, public and private hospital, and private practitioners. So trainers perform a face-to-face interview to eligible participants. Then we analyze the data using descriptive statistic and calculate the odds ratio using the logistic regression model. Okay, so before I continue to the next slide, I have a question for you. Okay. So
now we need the poll.
We need the poll, Sophia. So where do most TB patients go first when they have symptoms? Yeah. There is no right or wrong answer, but maybe you can tell us what do you think this patient mostly go first?
Hold on, I don't know what happened with the
the link.
The account, because it said someone logging from another device.
Oh, okay. Now we have the polls. Okay, community health center, private doctor, 2, 1, public hospital, private hospital. Okay, there's 7% for informal providers. Okay, almost there. For those who are not familiar, because here I can see there's some also Dutch participant. So the community health center is the primary healthcare unit that we have in Indonesia. And then of course, hospital, that's clear, I guess. And for the informal provider are those services that provide drugs like drugstore, pharmacy, or drug stockists, or maybe in another traditional healers, sort of like that. Okay, we can end the polling, Sophia. So most of them think patients go to community health center. And the second is public hospital and then private doctors. Okay. I think we can end the polling, right?
Okay,
okay?
Sorry. Yeah, okay.
Thank you for your participation. Okay, now we see. Sorry, the slide is a bit messy. So, I mean, here we can see that almost half of the patient actually go first to the informal provider. So they do self-medications by buying medications for their symptoms. And then only a small proportion go to hospital, and then the rest, like 30%, they went to the private practitioners. Yeah. Next slide. Okay. Now, and then another question. How many visits do you think is reasonable does a patient take on average to reach a TB diagnosis?
Can we have the poll?
Oh, yeah. Sophia. Another poll, please.
I don't know what happened with the poll. I actually made it, but then you are logging from another device and I cannot open the poll.
Okay.
So sorry, technical error.
Yeah.
Then we do it without the poll.
Yeah, we do it without the poll.
Maybe we can ask them to answer it on the chat.
Okay.
That's a bit complex.
Or maybe what you can do is just keep your answer and then we see. Oh, now it goes. Oh, no, this is the first. The poll is closed, but they don't have the second question yet.
Yeah, not yet.
Just go without the poll because otherwise we'll also run out of time. Continue.
Okay. Yes. So what we learned from the visits of the to TB diagnosis, it takes a median 6 visits for a patient until TB diagnosis. This is, of course, too long. So many of these patients had a very complex journey visiting both private and public provider before TB diagnosis. Next slide, Pak Renot. Okay, next slide, yeah. And with regard to the time to diagnosis, so starting from the onset to the initial presentation to the healthcare provider, it took a median of a month. This is quite long. And then from the first presentation to TB diagnosis, it takes another 23 days. So in terms of diagnostic delay, we have 62 days. And then for the treatment delay, it's not really a problem because it's only 1 day, but around 20% still start their medication after 1 day. So for the total delay, it looks like almost 2 months for a patient starting their symptoms until the treatment. And if we learn what are the risk factors for this, it's also quite interesting. Because we found that with regard to the patient's delay, male have a higher 1.6 higher risk to get a delay to the healthcare provider, and then lower education and no health insurance. So in Indonesia, since 2014, we have the JKN, the National Insurance Scheme. So this also, not having the insurance also make the patient probably higher delay compared to those who have the insurance. And also with regard to diagnostic delay, we found that those without insurance, of course, they will almost second time having delay than the one who have insurance, and then multiple visits, and then initial visit to a private practitioners. And with regard to treatment, of course, we also find multiple visits and then being diagnosed at the private practitioners also tends to have longer treatment delay. So as a summary, next slide, yeah. We can conclude that patient pathway in Indonesia are complex, involving both public and private sectors with multiple visits and long delays, especially to TB diagnosis. So a widely accurate diagnostic test could have a dramatic effect, reducing delays and onward transmission. So we would like to recommend to strengthen the public and private sectors, increase universal health insurance coverage, and also better access to diagnostic tools. Thank you. And I return to you, Pak Renot.
Thank you, Bonnie. Questions maybe via the chat, or we can have some questions at the end, but otherwise we'll move to a second brief talk, and that is by Raspati Kusumadinata, or Alid, who will speak about TB and diabetes. Over to you, Alid.
Thank you, Pak Renot. Hello everyone, my name is Raspati Kusumadinata and I'm here to discuss with you about tuberculosis and diabetes mellitus. So we will start with a question. Maybe Pak Renot, can you share the next slide please? And maybe we can try to poll, no?
Let's see if the poll works, Sylvie.
Let's see. Relaunch the poll.
So poll is in Mbak Raspati.
Yes, okay, I will try to.
I don't know what happened. It's moved to her.
No, sorry. It's a bit
This
difficult.
is the third one.
Yes, this is. Sorry. Maybe we just continue without the poll, yeah. So we start with a question. What is the most important risk factor for tuberculosis? The options are smoking, HIV, poverty, diabetes, and all correct. So I hope the participants already have their answers, and maybe we can just discuss. So tuberculosis, or TB, is an infectious disease caused by Mycobacterium tuberculosis, and it can affect all human organs, but mainly the lung. And there are a lot of risk factors for acquiring TB disease, and some are listed in the previous question. So smoking attributes to 20 to 30% of TB disease in high burden countries, including Indonesia. HIV, both high and low TB burden countries have attributed increasing TB incidence to HIV infection. Poverty related to malnutrition and overcrowding, both also important factors related to TB. And today we will discuss about TB and diabetes. Next slide, please, Pak Renot. Yeah. TB and diabetes are two important public health problems globally. In 2016, we see here there were 1.7 million deaths due to TB alone, and 95% of those deaths were in low and middle income countries, including Indonesia. Diabetes causes 4 million deaths annually, and the prevalence continues to increase. There are an estimated 80% people with diabetes in low and middle income countries, and 50% of them are undiagnosed. Next slide. A research collaboration between UNPAD and Radboud UMC on TB and diabetes started a long time ago, initiated by young Professor Reinoud van Grevel and Dr. Bakti Ali Shahbana. So when they noted that many TB patients in Indonesia also had diabetes, and most of them did not know that they had diabetes. In 2000, a paper on the association of TB and diabetes was published from this collaboration. Next slide. Studies on TB and diabetes sprung up around the globe. One recent systematic review confirmed that people with diabetes have 3 to 4-fold higher risk of acquiring TB compared with non-diabetes. And once they are infected, the treatment outcome is worse. Next, we have another question. Oh, sorry. Can anybody hear me now better?
Yes, we can hear you.
Yes.
Yes.
It's okay.
It's clear.
What are the effects of diabetes on TB? So this is a multiple choice. The poll seems to be working now. Can you see the poll? No?
Yes.
Yes, Alip.
Okay, but it seems that nobody yeah.
Nobody votes.
I don't know. I can't see.
It's not good for democracy.
Not good for democracy, yes. So maybe they know that, oh, they don't see the poll. They say that they don't see the polls.
I see. Okay. Well, then we continue.
Just a slide. Okay. Okay, so it seems that they vote through chats. Some people said chooses B, I think, and H, G, yeah,
E.
Yeah. So, so the answer to that question is that diabetes is associated with more active TB, more lung cavities, more drug resistance, more deaths, and more recurrence, but not TB outside the lungs. I don't think so, or the latent TB. Yeah. So actually, there's no correct answer on this one. If we have to choose only one answer, it's a tricky question. Next, Pak Reinoud. In 2012, our group initiated a multi-centered research project named Tandem, Tuberculosis and Diabetes Mellitus, with the aim to reveal the causal relationship between TB and diabetes, as well as to find better ways to screen TB in diabetes and vice versa, and to better manage patients with TB and diabetes comorbidity. Patients recruitment was done in Bandung, Indonesia, Peru, Romania, and South Africa, with collaboration with top universities in UK, the Netherlands, New Zealand, and Germany. Next. In Indonesia, we screened 809 diabetes patients for TB and 908 TB patients for diabetes. Among the combined TB and diabetes patients, we did a pragmatic trial on patients' management. This trial was supposed to be done in 3 countries, but Romania prohibited use of clinical diabetes algorithm in TB clinics, and Peru recommended the use of insulin in TB and diabetes patients. So we were only able to conduct this trial in Indonesia. Next. We wanted to answer this main research question, how to better manage diabetes in patients with active TB. We know that glycemic control is important but difficult for diabetes patients with moderate to severe infection, such as TB. And insulin is recommended but not widely available and expensive, at least in Indonesia. However, referring TB patients to diabetes clinics carries the risk of transmission to other diabetes patients who are healthy, and many patients prefer to be treated in their community health center closer to their homes. As a result, many TB diabetes patients receive inadequate diabetes treatment. So we developed an intervention combining intensified counseling, blood glucose monitoring, and diabetes medication adjustment, and we conducted a pragmatic trial to see whether the group of patients receiving the intervention would have better glycemic control at the end of their TB treatment compared to a control group. Next. We randomized TB diabetes patients we identified from the screening into 2 arms. As you can see here, intervention arm and control arm. And we collected data on their TB and diabetes status, physical examination, chest x-ray, and glycated hemoglobin or HbA1c at the beginning of their TB treatment, and then after 3 months, and then at the end of the treatment, that is after 6 months. HbA1c is the level of glucose in the red blood cells used to determine whether the blood glucose of the diabetes patients is well controlled or not. HbA1c below 7% is desirable, but diabetes patients with infection, the target is less stringent, it's 8%. Treatment and education for patients in the control arm relied on their treating physician in their original healthcare provider, while patients in the intervention arm were managed in our TB research clinic. Next. Next slide. Yeah. So we gave training to the patients in the intervention arm on how to measure their own blood glucose using a rapid test at home and write down the results in a diary. And they also should write down their TB and diabetes medication that they took every day. Next. We also developed an algorithm for adjusting medication. For example, as we see here, patients with a HbA1c less or equal to 10% at baseline receive metformin 2 times a day, that is flowchart A, and the dose can be adjusted after 1 week, while we also checked for side effects. If the target is not achieved, we can add insulin, this is flowchart B, or if there is a severe side effect, we go to flowchart C. Next. We also prepared flip charts and leaflet for counseling and education. Patients in the intervention arm were seen after 1 week, 2 weeks, and then monthly during their TB medication to receive the counseling. Next. So the results after 6 months of 150 patients randomized, 68 patients in the intervention arm and 66 patients in the control arm completed follow-up. Next. As we can see in this graph, the baseline HbA1c level of the two arms is similar, but after 3 months and 6 months, the HbA1c level decreased more in the intervention arm. And the proportion of patients who achieve the target level and the graph below of less than 8% is significantly higher in the intervention arm. Next. So this is one of our contribution. An international guideline on management of diabetes mellitus and TB was published in 2019. And here I show you next. This is one of the tables in the guideline, which is inspired with the work we've done in Indonesia. Two of the writers are our collaborator, Professor Julia Critchley and Professor Reinoud van Gravel. Next. In conclusion, we found in our trial that diabetes in TB patients were poorly controlled with baseline mean HbA1c around 11%. Therefore, we need to be more vigilant in screening for diabetes in TB patients and vice versa. And secondly, we showed that in a scientific study, a simple algorithm can improve glycemic control in TB clinic. Therefore, we need to develop a similar clinical algorithm which can be used more widely. And lastly, we recognize that the implementation of the algorithm under routine condition may be difficult, and we need larger studies to evaluate best approach and associate costs. Next. So these are several other questions we try to address in tandem, and some interesting other findings. We have some interesting other findings. For example, using whole genome sequencing, we found that Mycobacterial tuberculosis bacilli from diabetic patients carry a much higher number of genetic mutations that lead to resistance to TB drugs. This finding is published in European Respiratory Journal and was part of our friend Caroline Rusen's thesis. Next. Finally, I would like to thank our collaborators from UNPAD, our teachers from UNPAD, and then University of Otago, London, and Nijmegen, the Netherlands. Thank you. Back to you, Pak Reinoud.
Thank you very much. Thank you very much, Alid. The third topic is a bit more basic, a bit more lab focused. And I would suggest that maybe there's questions. We have a couple of questions via the chat now to Alid and Bonnie on this first part before we go to the molecular metabolomics and genetics for the final 15 minutes. Is there any specific, is there anything that people like to ask Bonnie or Alid regarding their studies on this patient pathway or the study on the diabetes? We can also do it at the end, but if there's a burning question now, please. Okay, in that case, we'll move to Edwin, who is more early in his PhD, because Bonnie and Alid will hopefully complete their thesis within 6 months from now. But Edwin will give us the final talk. Please, over to you.
Thank you, Parenot. Good morning, everyone. My name is Edwin Ardiansyah. I am now a PhD student starting last year on 2019, working on TB meningitis in omics. Next, Parenot. So TB meningitis is one of the most severe manifestation of TB, and if it often occur in small children and also in individuals with immunocompromised, like with HIV co-infections. And this disease has high mortality ranging from 30% to 50%. And our collaboration between UNPAD and Radboud has been going on, I think, for more than 10 years. And there are already 4 PhD graduated from these topics, 3 from Indonesia and 1 from Nijmegen. Next. Here we are seeing the MRI of patients with TB meningitis, and rather than the bacilli itself, the damage is mostly caused by the host immune response. Hence, we term it the term damaging inflammation. Next. So now we move to the poll, if it works. So the question is, amongst people infected with TB, what is the proportion of people getting TB meningitis? Yeah, so I think the poll is working, yes.
Well, for us it's working, but I'm not sure it's working for the audience. Otherwise, just vote by chat.
Yeah, but I can see
for the attendees, yeah.
Yeah, it's working. Yeah, so I see that 50% of the participants answer 1 to 2%. So, well, actually, it's quite difficult to estimate the number of cases, because sometimes the number of cases is underestimated. But according to WHO, it is indeed around 1 to 2% of people who is infected with TB get TB meningitis earlier or later in life. Yeah. And next, maybe. So what is the association between the inflammation and the mortality? Here we see that patients with higher body temperature, so higher fever, has higher mortality. So higher fever is associated with higher inflammation. And we also see that patients with lower CSF to blood glucose ratio also has higher mortality. And one of the important cells in immune response, the neutrophils, both in CSF and blood. So patients who have higher number of these cells in CSF and blood also have higher mortality. Next.
Edwin, have you mentioned what CSF means?
Oh yeah, I'm sorry. So for those who, maybe most of you are already familiar, but CSF stands for cerebrospinal fluid. So it's fluid that encapsulates the brain. And what about steroid treatment in TB meningitis? So a clinical trial conducted in Vietnam, randomizing patients receiving dexamethasone and placebo, showed that in general, steroid treatment decreased mortality, but the effect is more pronounced in the milder cases. And also the effect is mostly can be seen in the earlier years, and then it's over time it decreases. So next. So the big question is which biological pathway to target. So Dr. Rizal in Bandung, who already set up a cohort more than 10 years, collecting samples of CSF and plasma from TBM patients, but also from controls. So controls are patients who admitted with headache to the emergency, but we don't have an evidence of TB meningitis. And together with Clary Klish from Boston, they did a Metabolomix measurement. So Metabolomix is to measure the metabolites within the CSF. So those metabolites are the product of the brain metabolisms. And also together with Vinod Kumar, a geneticist in Nijmegen, and combining this clinical data, metabolomics data and genetics data, trying to figure out which pathway to target in TB meningitis. Next. So the sample is collected from CSF by lumbar puncture. Next. And then they did a comparison between patient who died and survived, and also between TBM and also control. Next. And they measured the metabolomics, which is the product of the brain metabolisms. Next. Yeah, so the analysis is done by Aryan from Nijmegen. What we see here is a volcano plot. So each dots represent metabolite, and everything on the right side is higher in TBM patient, and on the left side is higher in controls. And what we could see here, there are so many metabolites that are upregulated in TBM patients, but not so much in controls. Next. Yeah. So what about the comparison between patient who died and survivors? So this is also a metabolites plotting the ratio on the x-axis between the patient who died on the right side and on the left side. And on the y-axis is the ratio between survivors and the controls. So for example, if we took one example in the right top quadrant, LTB4, so this metabolites is in general higher in TBM compared in control, and also higher in patients who die compared to patients who live. Next. So the opposite on the left corner quadrant, for example, the glucose is the opposite. So it's higher in controls and it's lower in TBM, and it's higher in patients who are alive and lower in patients who died. Next. And we see one dot on the right corner, which is very interesting because looking at the ratio, it is almost 10-fold higher in patient who died. And that dot is tryptophan, which is higher in controls compared to TBM patients. But in TBM patients itself, it's slightly higher compared to the survivors. Next. Okay, so the discovery, it was done on a set of 55 patients, and this effect is validated in a larger group of patients of 124. And then if we divided the patients stratified by their CSF tryptophan level, so patients with higher tryptophan level on the light gray and low tryptophan level on the black line, we see that patients with higher CSF tryptophan level indeed have higher mortality, so lower survival. Next. And why would we think that tryptophan is relevant? So some possible explanations, including first, the tryptophan itself is a nutrient for the mycobacterium. And second, some of the tryptophan metabolites can modulate the immune response. And third, some of the product of the tryptophan degradation have some neuroprotective effect or neurodamaging effect. Next. And here we are looking at the tryptophan pathway. So tryptophan in the circle, and 90% of the tryptophan flow into the kynurenine pathway, which is below. And one of the important enzyme that degrade the tryptophan called the IDO1. And in one paper that compares the gene expression in the brain of TBM patients and patients who died because of accident, we see that IDO one gene is expressed in TBM patients, but not in patients who die because of trauma. Next. And then can we support this with genetics? So is the variability in CSF tryptophan levels also controlled by genetics? So instead of going by candidate gene, for example, if looking into the polymorphism within the IDO gene and look into those genotype, we go unbiased. Unbiased meaning we screen the whole genome by SNP typing. So we genotype around more than 400,000 SNPs within the genome. And then we do imputation. So imputation is prediction based on the known SNPs. And in the end, we have 6 million to 8 million position of SNPs, the genotype. And what we do is we try to associate the genotype with the phenotype, in this case, the tryptophan. And we try to find association between the genotypes and the trait. And we are using a continuous trait, so the tryptophan is continuous trait, and the benefit is it increased the statistical power compared if we are using a binary trait, so like a disease versus healthy. So hoping we could detect effect with smaller number of samples. Yeah. And the results is typically plot and a plot like, yeah, I hope everyone can follow. So the result is usually plot in a plot called the Manhattan plot, like this. So every dot resembles one SNP, and it is ordered by their position across the genome, and also ordered by the chromosome number. And in the y-axis is just the statistical significance in minus log scale. So the higher the scales in the y-axis, the more significant. And then we have the blue line, which is the threshold line. So everything above that line is significant. So for example, in chromosome 7, we have one dot that is above the line. And if we look into that genotype, that we can see that patients with TB genotype has higher CSF tryptophan level compared with a patient who have AA genotype. Next. And in this QTL result, we see 11 different loci in the genome that are associated with the CSF tryptophan level. And using these 11 genotype, we can somehow combine them and calculate a score. We call it the genetic risk score. So every patient can have a score based on the genotype of this 11 position in their genome. And then if we stratified patients by their scores, the patient with high and low scores, we can indeed see that patients with higher genetic scores have higher mortality. And then this is validated in another set of 285 patients who have genetics but do not have tryptophan measurement, and we can validate the results. And then what about other metabolites? Here on the left side, we can see that other than tryptophan, there are also many metabolites that are upregulated in patients who died. And apart from the metabolites that we already know the identity, we also have a lot of thousands of measurements that we know the values, but we don't know the identity of the metabolites. And if we do the similar comparison, we found 190 metabolites that are different between patients who died and alive, but only 15% of them already annotated, or we already know which metabolites are that. So there are many still unticked metabolites. So now this comes to the ultimate project. Okay, which is a project ongoing starting last year, which tried to took the same approach, but with larger group number of patients from Indonesia, but also now in Vietnam. So we want to see if there's a similar pattern within the metabolites, also the genetics. Yeah, so for conclusion, omics study can provide insight into biological pathway involved in immunopathology and TBM. And we see that cerebral tryptophan is important in mortality in TBM, but also we also see many other metabolites involved. And we will do whole genome sequencing in addition to the SNP genotyping, and to hopefully develop a host-directed therapy for TB meningitis. Thank you very much.
Thank you, Edwin, for this nice story. There's a few questions from the chat, so the first question, and maybe brief answer to Alit: what is the main difference in medication algorithm between control and intervention arm? And was the modification limited to the management of diabetes? And did you also look at DM parameters besides HbA1c?
Yeah, I think the last question is revised. She wanted to ask whether we also look at TB parameters. Yeah, so for the first question, the difference of medication. So this is actually a pragmatic trial. So it's not really a controlled clinical trial. So the intervention arm were treated in our TB research clinic with the intervention package that I've described. And the control arm, they were returned to their original healthcare provider. So the story is we asked the community health center to send their patients to us, their TB patients or DM patients, to be screened. And then when they are eligible, then we offer them to be enrolled in this trial. So when they're randomized into the control arm, we refer them back to their original healthcare provider. So they could be referred back to the puskesmas, the healthcare, or hospital that they were referred before, and then yeah. And so maybe if I just short this, so usually in the puskesmas they get metformin and sulfonylurea, but in our and rarely get insulin. And yes, we limited to DM management because the TB management is already actually follow the dots. And then we did look at TB parameters such as sputum conversion and all, but we think that the sample size is not enough to detect any difference, and it's not, so we didn't look at it. And
thank you. There's one more question for you, Alit, and that is involvement of policymakers. Or
yes. Yes, we work closely with the City Health Office in Bandung because we need their permission to recruit patients from the puskesmas. And we also work closely with endocrinologists, so they also be informed of our results. And some
good. I'm going to cut you short, Alit.
Yeah, okay.
Because there's also a question for Dr. Boni, and that's about whether the COVID-19, how it would affect your pathway.
Yes, thank you for the question. I don't have the figure for Indonesia, Ibu Margaret, but in West Java, yes, we have a decline in the number of TB cases compared in 2019. Since April, we record that less than half of, so the case detection rate is half less than in 2019. That's what I can tell. And we don't have structured research for that. With regard to the case detection, yes, it is indeed decreased in 2020 after COVID.
Excellent. So we have a couple of minutes left. Obviously, the topic by Edwin is very complex. It's published. It's a beautiful paper. You can find it on the tryptophan and the TB meningitis, like many of our other work. I would just like to share, because it's not just about the content of the science, but just what I think makes this academic collaboration special. I think one is that we have sort of a comprehensive approach. So we looked at tryptophan as a molecule, we looked at a TB diabetes patient in the clinic, and we look at a population at the health systems. And there is a complementary expertise. And as you've seen today, there's a lot of talent in this group, which is also very well embedded and engaging with policymakers, etc. And of course, the international collaboration and international funding is essential. We have long projects, which helps a lot. And finally, but definitely not very important as well, is that with Dr. Bhakti and Prof. Nina and others, we have very strong leadership in the Indonesian, in this Bandung TB group, and also strong values, let's say, of the things we want to achieve collectively. And also, I think what is important, there's a lot of fun. So with that, I want to thank, and this slide could contain many, many more people, but some of the people involved in this collaboration. And like to thank you and for your attention, and of course the other speakers for their contribution to this session, which I hope you enjoyed.